Hello, I am Ashish Arora. I am working as a senior scientist at the Molecular and Structural Biology Division of CSIR Central Drug Research Institute at Lucknow. Here I present to you under the EPG Pathshala, Paper 8 Medical Physics, Module 28, which is Methods of Multidimensional NMR for Biomolecular Structure Determination, Part 2. The objectives of this module are to present to you how the aliphatic and aromatic side chains are assigned, what are the 3D toxi experiments for side chain assignments of aliphatic residues, what 2D and 3D NMR experiments are used for the assignment of aromatic residues, how dihedral angle constraints are generated from the assigned chemical shifts, what are the 3D Nozi Hsqc experiments to map the NOEs between protons, how is the protein structure calculated from the constraints, and how is the protein structure refined, validated and submitted to Protein Data Bank. In our previous module, we have learnt about the experiments that provide backbone resonance assignment through sequential walk. Now let us look at the side chain atoms of amino acids. These are taken from the Cyana library, and in this the various amino acid structures are shown and the specific labeling for their atoms are also given. Let us look at some of them. For example, at the right lower corner we have valine. So, valine has your C alpha linked to H alpha, which is not shown, and then in the side chain you have C beta, which is linked to H beta, and then you have C gamma 1, which has 3 protons labeled as Hg11, Hg12 and Hg13, and then the other methyl group which is labeled carbon as Cg2 and with protons as Hg21, Hg22, Hg23. We have serine, in which we have Cb, which has Hb2 and Hb3, and this has a hydroxyl group OG and Hg. Then we can look at interesting residue proline. There is the N, and N has the C alpha. There is no NH in proline. N has C alpha, which has an H alpha, and the C alpha is connected to carbonyl, also to C beta, which has Hb2, Hb3, then Cg to Hg2, Hg3, and C delta or Cd to Hd2 and Hd3. Then C delta closes on the nitrogen. We have the simple glycine residue. It has Ca and its 2 hydrogens are labeled as Ha2 and Ha3. Let us look at a longer residue, isoleucine. So, in isoleucine, we have isomerization with respect to leucine. We see Cb, which has 1 Hb, then we see methyl group linked to C beta as Cg2 and the hydrogens connected to it as Hg22, Hg21 and Hg23. Then there is a second carbon connected to Cb, which is Cg1, and the hydrogens are Hg21 and Hg22. And then we have a C delta methyl group, and it has HD11, HD12 and HD13, and so on and so forth for other residues. One can see that there are hydroxyl groups present in serine and threonine, which would give a very typical C beta chemical shift, and then there is a methyl group present in alanine, which again has a very typical chemical shift, and then there are certain other features for other residues. Side chain assignments. Backbone assignment experiments provide the chemical shift of Hn, N, C alpha, C beta and the carbonyl CO only. We can have an experiment called as HB, HA, CO, NH. This experiment would provide the chemical shifts for the HB and HA just like the CB, CA, CO, NH for the preceding residue HB and HA through the amide NH, and this is a one directional experiment. The remaining chemical shifts to be assigned for various residues are H alpha and H beta for amino acids except for glycine, C gamma, H gamma for valine, isoleucine, leucine, glutamic acid, glutamine, methionine, lysine, arginine, proline and threonine, C delta and H delta for isoleucine, leucine, lysine, arginine and proline. Then you have C epsilon and H epsilon for lysine and methionine. Side chain amide N and H for asparagine and glutamine. Side chain NZ and HZ for lysine and N epsilon and H epsilon for arginine. Aromatic side chain hydrogens for histidine, tyrosine and phenylalanine. Let us look at their labeling. Histidine C beta is linked to HB2 and HB3. CB is linked to CG. CG is linked to CD2 which is linked to a hydrogen HD2. CD2 is linked to NE2. Ne2 to Ce1 which has the hydrogen He1, and then Ce1 is linked to Nd1 which has hydrogen Hd1. So, basically for histidine in the aromatic side chain we can characterize the chemical shift of He1 and Hd2 protons. Now, let us look at the experiments for the assignment of side chain atoms. The side chain chemical shifts of residues are assigned from the following experiments: H bracket CCO bracket closed NH toxi bracket H bracket close CC bracket CO bracket closed NH toxi HCCH toxi C13 proton Hsqc for the aliphatic region and same for the aromatic region. Then 2D bracket HB bracket closed CB bracket CG CD bracket closed HD for aromatic side chains, and the same for the HE for aromatic side chains. Let me explain this a little. As I mentioned earlier, whatever atoms are mentioned inside the brackets, their chemical shifts are not recorded, they are not labeled. So, when we have HCCONH toxi experiment, we are labeling only the proton of the side chain, and when we have bracket H bracket closed CCCONH toxi experiment, we are labeling the carbon chemical shifts, and we will learn about these experiments individually in the following slides. Let us start with the 3D HCCONH toxi experiment. This experiment is used to assign aliphatic side chain protons. The side chain protons are frequency labeled. Magnetization is transferred from the side chain hydrogen nuclei to the attached C13 nuclei. Then isotropic C13 mixing is used to transfer magnetization between the carbon nuclei. We have learnt about the proton-proton toxi, and here there is a C13 toxi. From here, magnetization is transferred to the carbonyl carbon onto the amide nitrogen and finally the amide hydrogen for detection. The spectrum correlates the Hi, Ni from the amide group with proton of side chain of the residue that is preceding, that is I-1 from the side chain of the preceding residue. It is especially useful for assigning the side chains of proline residues. And in this figure, we have strip plots showing assignment of side chain hydrogens of segment L32 to I36 of CEGMF. CEGMF is the C. elegans glia maturation factor, which was described in the previous module. And in the previous module, it was also shown that a 3D spectrum is broken down into strips by first taking a plane out at the nitrogen chemical shift and then making a strip at the with proton chemical shift at the center. So, here we see the strips from L32, I33, L34, K35 and I36, and on the left we see a schematic for the Hsqc and Hsqc experiment. In red you can see that Hn is linked to its own N, and then from N to the preceding carbonyl, from the preceding carbonyl to the C alpha, C betas and C gammas, so that you have the carbon toxi, and then from there you have the H alpha, H beta and H gamma. So, basically as the arrows point out, magnetization starts at the side chain protons, it is transferred to the respective directly linked carbons, and from the carbons it goes to the carbonyl, from the carbonyl to N and then to NH. And now as you can see in the strip I36, so I36 will give you for K35 the various proton chemical shifts for the side chain. And similarly for K35 you have L34, for L34 you will have the peaks of I33 in the strip of L34 and so on and so forth. Now, the simplest one that one would want to see is the strip of L32, which shows you the proton chemical shift of the preceding residue, which is alanine 31. Alanine, as we know, has HA and HB. HB comes from the methyl group, which is attached to C alpha. And we see that the proton of this, it has a chemical shift close to 0.8 ppm, whereas the H alpha chemical shift is around 5.6 ppm. So, this is the simplest proton chemical shift for the alanine side chains. At the same time, you can see in the strip of I36 the complex spectrum with many peaks which correspond to the proton of the side chain of K35, which are shown as very clear. At the bottom we have HAK35, then we have the HE2 and HE3 of K35, which have a lower, which have actually a higher chemical shift because they are attached to a electronegative atom, and above them we have HB2, and then we have the HD2, D3, and then at the top we have HG2 from K35. Now, we see the spectrum 3D bracket H, Hsqc, CO and Hsqc. This experiment is used to assign aliphatic side chain carbons. Magnetization is transferred from the side chain hydrogen nuclei to their attached C13 nuclei. Then isotropic C13 mixing is used to transfer magnetization between the carbon nuclei. The side chain carbons are frequency labeled. From here, magnetization is transferred to the carbonyl carbon onto the amide nitrogen and finally the amide hydrogen for detection. This spectrum correlates the HI-NI from the amide group with carbon side chain of I-1 from the side chain of the preceding residue. It is specially useful for assigning the side chain carbons of proline residues. Now, in this experiment, again what you see are the HCCONH toxi strip plot showing assignment of side chain hydrogens of segment L32 to I36 of CEGMF. On the left side, you see the magnetization transfer. The only difference from the previous experiment is that the magnetization again starts at the protons and it is transferred to the directly attached carbons and an isotropic mixing sequence is applied. But then after that, it is the carbon chemical shifts that are labeled in place of the proton chemical shifts that were labeled in the previous experiments. The rest of the experiment is same. Magnetization is transferred to carbonyl and then to its directly attached N, and from there to Hn, and that is where it is detected. Again, for simplicity, now let us look at the strip of L32, which is taken at a nitrogen chemical shift of 125.64 ppm and a proton chemical shift of 8.94 ppm. This strip shows two peaks. The peak at the downfield peak, which is at the bottom, is labeled as Ca of A31, which is now the preceding residue to L32. So, this is the C alpha chemical shift, and this would match in the HNCA and HNCA CB experiments for the strip of A31. And we also see an upfield peak, which is at a very characteristic value of around 22 ppm, and this is of the C beta carbon chemical shift of A31. So, this experiment provides you with enough peaks to kind of reconfirm the assignments that are made in the HNCA and HNCA-CB and CBCA-CONH experiments. You see a more complex pattern for I36, which would show you the carbon chemical shifts of the preceding residue, which is K35. And as you can see in this strip, there is Ca K35, Cb K35 and Cg K35 labeled, whereas others are not labeled because of clarity. Then there are other residues, and it is interesting to look at them. For example, if we look at the strip of L34, then what we see is that this is taken at a nitrogen chemical shift of 128.24 and a proton chemical shift of 9.54. It shows the preceding Ca of I33, then on top you have C beta of I33, and then you have the Cg1 and Cg2, and also you see Cd1 of I33. So, that is how the carbon chemical shifts of side chain are assigned. Now, 3D Hsqc toxi. This experiment yields strip at each carbon frequency in the side chain, in which all side chain proton resonances are visible. Magnetization is transferred from the side chain hydrogen nuclei to their attached C13 nuclei. This is followed by isotropic C13 mixing, and finally magnetization transfers back to the side chain hydrogen atoms for detection. Both the side chain protons and carbons are frequency labeled. This experiment helps to correlate the chemical shift information obtained from 3D HCCONH toxi and 3D bracket H bracket closed CCCONH toxi. In this slide, we see HCCH toxi strip plot showing proton resonances in the plane of each carbon of D33 residue of CEGMF. So, there are 3 strips, and these 3 strips are interrelated, or rather intra-related. All of them, they have different chemical shifts. You have the left strip with a carbon chemical shift of 53.45, which corresponds to the C alpha carbon, and then you have the C beta carbons at 44.46, and then there are these proton chemical shift for the C alpha. The H alpha is at 4.88, whereas the others are at 2.72 and 2.22. So, here what you see is that in the left strip, you have at the bottom labeled as HAD33. So, a combination of CAD33, HAD33 gives you the diagonal peak in HCCH toxi, and this diagonal peak has 2 cross peaks, and these cross peaks are labeled as HB2 D33 and HB3 D33. Now, in the next strip, what we see is that it is a bit crowded, but in the center we see C beta linked to H beta, and this is what forms the diagonal, and then this diagonal has two cross peaks, HB3 of D33 and HA of D33, and in the right strip now the diagonal is formed by C beta and HB3, which is right at the top, and then this shows cross peaks with HB2 D33 and HA D33. Sometimes what happens is that when a CG or a CD has two protons attached to them, or there are two CGs, then it is difficult to tell which proton chemical shift corresponds to which carbon chemical shifts, and this is clarified by looking at the diagonal and cross peaks in the HCCH Toxi strip plot. Now we have the two-dimensional C13 proton Hsqc showing the peaks for the aliphatic region. Many of the very sharp peaks are labeled as HD1, CD1 for I-123 and so on and so forth, and these basically the sharper peaks come from the methyl groups. Then you have other peaks that come from the C beta, H betas correlation, and then in the region from 3.5 to 5 ppm you usually have C alpha, H alpha correlations. So this is what the 2D C13 proton Hsqc spectrum looks for the aliphatic region. Now we look at the 2D bracket HB bracket closed CBHD, which is simply called as CBHD experiment, and likewise a 2D CBHE experiment. These are two-dimensional experiments and are used to provide C13 beta to 1H delta and C13 beta to 1H epsilon correlations. These experiments can be performed with either water or deuterium oxide samples with good sensitivity. They provide a simple approach for the unambiguous sequence-specific assignment of aromatic side chains. The path of magnetization transfer for 2D CBHD experiment is from H beta, the signal goes to C beta, then to C gamma, then to C delta, and then from C delta to H delta. The chemical shifts are labeled for C beta and for H delta. That is why it is called as a CBHD experiment. And similarly, the path of magnetization transfer for 2D HB, CB, CB, CG, CD, HE is that the magnetization goes from H beta to C beta, then from there to C gamma, to C delta, to C epsilon, and then to H epsilon. This is a longer experiment and it is less sensitive than the CBHD experiments. As I was talking about the side chain of the histidine residue, this experiment can provide the C-B correlation with the HD and HE of the histidine residue. So here you see the CBHD spectrum of CEGMF with various correlations that are seen between the aromatic C beta chemical shifts and their HD protons in the aromatic side chain. And similarly, you would see for the C beta HE experiment. Now the extent of assignment and data deposition. After assignment, all the assigned chemical shifts in a protein are deposited with the Biological Magnetic Resonance Bank BMRB, and its website is given. This is hosted at the Wisconsin University. Assigned chemical shift data must be given in the NMR star format for the BMRB deposition. I just want to share with you that I have done my postdoc in USA at the University of Virginia and had a very close collaboration with the University of Wisconsin at Madison, and this BMRB is hosted by Dr. John Markley in his group. I worked for some time and I have published a paper in Nature Structural Biology on the structure of outer membrane protein A, transmembrane domain, in collaboration with this group at the BMRB at Wisconsin University. The chemical shifts of C. elegans GMF have been deposited in the BMRB under accession number 36128. Backbone resonance assignments were made for 130 residues out of 132 possible non-proline residues. Assignments for 99.3% of C alpha, C beta and carbonyls, 97% of non-aromatic and non-carboxyl side chain carbons, 53% of aromatic side chain carbon and 86% of aliphatic side chain hydrogens and 66% of aromatic side chain hydrogens were completed. So, here in this figure what you see is the same N15 proton Hsqc spectrum of N15 labeled CEGMF, and this shows you assigned residue names and numbers. For example, G13 is the amide NH peak for glycine at position 13, same for G95, then B14 that you see at the lower end on the right side is of valine 14 and so on and so forth. So, by this analysis, various backbone assignments, we are now able to assign the amide NH peaks to their respective positions in the sequence. The peaks in green are the side chain NH2s and they are labeled, for example, He21 and Ne2 of Q44 and He22, Ne2 of Q44 and so on and so forth. Identification of secondary structural constraints from chemical shifts. Prediction of secondary structure, elements and constraints from chemical shifts is done by using the software Talos Plus. It can also be done with CSI and some other softwares. Talos Plus incorporates a neural network classification scheme and a large database of 200 proteins. Talos Plus can, on an average, make dihedral angle predictions for approximately 88% of residues in a given protein. Talos Plus breaks the sequence of a target protein into overlapping amino acid triplets. For each triplet, the program searches its database for high-resolution X-ray crystal structures to compare the chemical shift and sequence similarity. The torsion angles for the central residue from the best 10 matches are chosen as the predicted torsion angles. Then you see is a figure. This figure is the residue specific torsion angle predictions based on which the beta strands and alpha helices are identified along the sequence. So, this gives you the prediction of secondary structure elements from Talos Plus for C. elegans glia maturation factor. As you can see, it starts with beta strand and they are shown in green, and then on the other end you have the alpha helix 1, then you have strand 2 and strand 3, you have helix 2, then you have strand 4, strand 5, strand 6 and strand 7, which is followed by the long helix alpha 3, and then you have the strand 8 and with a C-terminal alpha helix. These are all predictions of secondary structure from Talos Plus program. NOE assignment and interproton distance constraints from NOE. We have read quite a bit about nuclear over-Auger effect and Nozi. Nuclear over-Auger effect measures the distance between two dipolarly coupled spins and hence provides interatomic distances or distance information for protein structure determination. NOEs provide short range 1.8 to 2.5 angstrom, medium range 1.8 to 3.5 angstrom and long range 1.8 to 5.0 angstrom distance information between pairs of hydrogen atoms separated by less than 5 angstrom. The short range NOEs, D alpha N I plus 1, DNNI plus 1 and D beta NI plus 1 and medium range DNNI plus 2, D alpha NI plus 3 and D beta NI plus 3 NOEs are valuable for defining secondary structure elements such as alpha helix or beta sheet. The long range NOEs provide crucial tertiary structural information. The routinely used NMR experiments to obtain NOE connectivities in proteins are C13 or N15 edited 3-dimensional Nozi Hsqc experiments. The short and medium range NOEs for different secondary structures are shown in this table, shown as a figure in this slide. So, the distances are shown as D alpha N, D alpha NII plus 2, alpha NII plus 3, D alpha NII plus 4, then DNN, DNNII plus 2, D beta N and D alpha beta I plus 3. So, as you can see, they are given for the alpha helix, for 310 helix, for the beta antiparallel and beta parallel strands and sheets, and they are also given for the type 1 turn and the type 2 turn. You should have a look at these distances so that you know what kind of NOEs can be observed, and a complete set of NOE is very helpful in assigning the right secondary structure. Let us now look at the N15 edited Nozi Hsqc experiment. In this 3-dimensional experiment, magnetization is exchanged between all hydrogens within 5 angstrom distance through the NOE. Then the magnetization is transferred to neighboring N15 nuclei and back to proton for detection. So, this is the Hsqc component. Usually a mixing time of 100 to 150 millisecond is used for the generation of NOE. All protons in the vicinity of 5 angstrom of any amide proton are detected in terms of NOE cross peaks to that amide proton. The integrated intensities are converted into distance constraints by using various modules of the Cyana software. In this slide, we see strips from the L103 to S103 segment of N15 edited Nozi Hsqc. This slide has been tilted by 90 degree to give you a better idea of the resolution or separation of the peaks. On the right side, you have labeling of N15 ppm and you have the residue numbers L103, Y104, A105, G106 and S107. Below the residue number, you have the nitrogen chemical shifts where these peaks were taken at, and then right at the bottom on the left side, you have the proton chemical shifts. So, basically what you see, and there are various labels on the peaks, what you see here that there will be a strong peak at the bottom, which will be the Hn of that particular residue, and that would be the diagonal peak. So, Hn, wherever the peak is labeled as Hn for the same residue number, for example, for alanine 105, the H or Hn 105 is the diagonal peak right at the bottom. And then all the other peaks are basically the peaks of protons that are within 5 angstrom distance. And if you try to count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, you can count up to like 20 to 25 protons that are close to the amide Hn for the A105 residue. And these peaks are then converted, their intensities are converted into distance, and that is how you get distance constraints. Some of these peaks come from the same residue, some come from the adjacent residue, some comes from close residue in the secondary structure, whereas many of them are long range NOEs, and this helps to work out the tertiary structure and fold of the molecule. C13 aliphatic and C13 aromatic edited NOE Hsqc. These are two different experiments. These spectra are used to obtain restraints from hydrogens associated with aliphatic and aromatic carbons. it is usually recorded with 100% D2O as the solvent to minimize the artifacts from exchangeable protons. Generally, amides are exchangeable, or amide protons are exchangeable. Therefore, when you put the sample, when you put D2O in the sample, you actually lyophilize the sample and rehydrate it with 100% D2O or 100% D2O buffer. Then what happens is that the amide NH get exchanged, and so they do not give any spurious peaks in the spectrum, so that the spectral width can be shortened and one can only look for the CH aliphatic region or with the CH aliphatic to CH aromatic. For C13 aliphatic edited Nozi Hsqc, the transmitter offset for carbon is centered for the aliphatic region, which is around 45 ppm with a spectral width of 70 to 80 ppm for carbon. By the way, for N15 edited Nozi Hsqc, the N15 transmitter is usually kept at 120 ppm nitrogen chemical shift. For C13 aromatic edited Nozi Hsqc, the transmitter offset for carbon is centered for the aromatic region at 125 ppm, and it has a shorter spectral width of 40 ppm for carbon. The integrated intensities are converted into distance constraints by using various modules of Cyana. So, here in this figure, we see the strips from the 3D C13 edited Nozi Hsqc. On the left, you see from the aliphatic region, and on the right, you see from the aromatic region. That is why the proton chemical shifts on the left are less than 4.5 ppm and they are more than 4.5 ppm for the strips shown on the right. The right residue is phenylalanine 57, whereas the residue shown on the left for aliphatic strips is the threonine 28. So, if we look carefully, then we see that this T28, there are various carbons corresponding to this residue at 60.81 ppm, at 69.93 ppm and at 22.54 ppm. And then there are protons attached to them. We have HA, we have HB and HG protons also, and now these protons give NOEs with different other, so that we have short range and long range. For example, we can see that in the third strip for T28, that there is the NOE correlation of this with HB3 and HB2 of residue D51. Hydrogen bond constraints. Hydrogen bonds can significantly improve the structure quality and are usually incorporated into the secondary structures. Hydrogen bonds are usually incorporated into the secondary structures as predicted by Talos Plus and confirmed with characteristic NOE patterns and experimental hydrogen deuterium exchange. Amide protons participating in hydrogen bonds can be determined by rapidly dissolving the lyophilized protein in D2O solution, followed by repeated acquiring of N15 proton Hsqc or homonuclear toxi spectra. Specificity of hydrogen bond constraints are based on the distance 1.8 to 2.1 angstrom between the donor and acceptor atoms. A total of 28 hydrogen bonds were added in CEGMF for structure calculations. Structure calculation and its refinement. For three-dimensional structure calculation, distance constraints derived from NOE assignment, dihedral angle constraints derived from chemical shift based Talos Plus predictions, and hydrogen bond constraints derived on the basis of HD exchange are used as input. There are several computing algorithms for structural calculation which are based on the distance geometry, restrained molecular dynamics, torsion angle dynamics and simulated annealing. Cyana is the most popular one, which is based on torsion angle dynamics protocol. A distance restraints file is generated using peak file, atom list file and sequence file. In the next step, structural calculation is done and PDB file .pdb and overview file .ovw are generated. Repeated cycles of cyana are run to remove violations and to reduce target function. Finally, 100 conformers are generated and 10 to 20 lowest energy conformers are selected. The final 10 conformers are subjected to explicit water refinement using the CNS 1.21 program. And doing all this process, we arrive at an ensemble of structures, and this ensemble of structures representing the NMR solution structure of C. elegans GMF is shown in the figure as A and also as B. In A it is shown as a ribbon diagram and in B it is shown as the backbone traces. This structure has been deposited in PDB and has ID of 5YNR. You can see in red are the alpha helices and in cyan are the various sheets. Next. So, the NMR structure is an ensemble of structure solutions that come from the structure calculating program. There is not a single unique structure that would describe the NMR solution structure of a protein. So, the NMR structure carries with it inherently the information about the disordered regions, the less ordered regions, the dynamic regions and also correspondingly the rigid regions in the remaining scaffold of the protein. Structural parameters for final ensemble of 10 structures of CEGMF, and these parameters have to be provided to the journal. These come directly from the validating softwares. So, completeness of resonance assignments: backbone 97%, side chain 96% and aromatic 84%. Conformationally restricting restraints, you have distance restraint list, a total you have, and for CEGMF you have the intra-residue 464, sequential you have 699, medium range 886 and long range 1092. There are 28 hydrogen bonds. There are dihedral angle restraints phi and psi amounting to 224. There are no disulfide restraints because there are no disulfides in the molecule. So, number of restraints per residue on an average is 22.76 and number of long range restraints per residue is 7.91. Structure validation and deposition. The final set of structures which have the lowest conformational energy are selected. The quality of these structures is analyzed using softwares such as ProCheck, Psvs and What If. Psvs provides numerous structure quality scores for Verify 3D, PROSA, ProCheck, Sci-Fi, ProCheck All and mole probability clash score. Z-scores for ProCheck and mole probability of -2 and above are desirable for a high quality protein NMR structure. One should never deposit a structure to the PDB with Z-scores of less than -4 or more than +4 for ProCheck scores and mole probability clash scores. Validation of CEGMF structure by Psvs. You have the residual restraint violations, average number of distance violations per structure in the range of 0.1 to 0.2 angstrom, 0, 0.2 to 0.5 angstrom, 0, 0.5 to or greater than 0.5 angstrom or also 0. So, a good calculated structure should not violate the restricting or the constraints, NOE constraints that one has given, and when the constraints are violated, these are called as the distance NOE or NOE violations or distance constraint violations. And the dihedral angle violations in the range of 1 to 10 degrees is also 0, greater than 10 degrees is also 0. Model quality, you have RMSD, backbone atoms is 0.5 and for all heavy atoms is 0.9, but this is for the selected secondary structure regions. Bond lengths, you have RMSD of 0.005 angstrom and bond angles you have of 0.7 degrees. And this mole probability Ramachandran statistics, most favor regions are 97.2%, the allowed regions are 2.6% and additionally allowed regions are 0.2%, so that the disallowed region are 0%. And the quality scores, verify 3D is 0.38 / minus 1.28, ProCheck 5 psi minus 0.13 minus 0.20, and model contents, total number of residues is 138, the BMRB accession number is 36128 and PDB ID code is 5YNR. With this, we come to the end of this module and let us summarize. For high resolution structure, side chain proton, carbon and nitrogen chemical shifts have to be assigned. The aliphatic side chain chemical shifts are assigned through the 3D Toxi type experiments like HCCONH Toxi, CCCONH Toxi and HCCH Toxi. The aromatic side chain chemical shifts are assigned through 2D C13 proton Hsqc aromatic and 2D CBHD and CBHE type of experiments. Dihedral angle constraints are predicted from the chemical shift through the program TALOS Plus. Distance constraints are generated by assigning the NOEs in 3D C13 or N15 edited Nozi Hsqc experiments. Hydrogen bond constraints are verified by HD exchange experiments. All the constraints are put into a software like Cyana or CNS or NIH Explore to calculate structures. Approximately 100 structure solutions are generated and 10 with lowest target function are chosen to represent the solution structure after explicit water refinement by CNS and validation by Psvs. The final ensemble is deposited in PDB and analyzed for various purposes. Thank you.